In this part, we'll be looking at receptors and how we can modulate receptor activity. So within this part, we'll be looking at the structure and function of different receptor types, including ion channels, G-protein coupled receptors, or GPCRs, and kinase-linked receptors. And in future sections, we'll be looking at the chemical design and function of agonists, antagonists, partial agonists, and inverse agonists as well. So what is a receptor? Well, a receptor is a protein molecule that's embedded within the cellular membrane with part of its structure on the outside of the cell. So it's a structure that bridges the cellular membrane and allows messages to be sent from outside of the cell to the inside of the cell. Now, receptors are absolutely crucial for different communication pathways within the body. Now, neurons carry messages from the central nervous system, but they stop short of the target cell. So a neuron doesn't actually carry the message into the target cell itself. It allows for long-range transmission of a signal, but it doesn't actually give that signal into the cell. That is done by a process involving receptors. Now, what we can see here is we have our nerve cell, which carries a signal down to the end of the cell. When this signal reaches the end, a messenger molecule is released, and this traverses the gap between the end of the nerve and the receptor on the cell surface. Now, when this messenger enters into a receptor on the cell surface, this transmits the message that is being carried into the cell, and it forms parts or initiates whatever the cellular response mechanism to that message might be. So this process of carrying the message from the end of the nerve into the cell is performed by a chemical messenger, and it's this chemical messenger that binds to the receptor and initiates the cascade of events within the cell that results in the required action. Now, here is another example where we could have in our blood supply, we have hormones, which are chemical messengers. Now, these hormones can also interact with the surface of the cell. It can also interact with receptors, and a cell can receive messages from many different sources, including from hormones within the blood to nerve endings as well that happen to terminate close to that particular cell or cell type. And it's this combination of hormones and neurotransmitters that allow us to get messages into cells and tell cells when they need to mount a biological response to something, perhaps to some sort of external stimulus. So receptors really are key in permitting the transmission of a message from outside of a cell to the inside of a cell. And in this section, we're going to have a look at the structure of different types of receptors, and then we'll look at how we can both enable this message to be sent and also stop this message from being sent as well when we look at agonists and antagonists in future sections. Now, I just wanted to show you what the chemical structure of a receptor actually looks like. Now, receptors are all protein molecules, and a lot of receptors have a significant portion of alpha helices associated with them. Now, on this diagram, you can see this here as the red helices. These are all alpha helices. Now, the reason they have this is because they transverse the cellular membrane. And so receptors that cross the cellular membrane often have a lot of alpha helices associated with them. Now, this particular receptor is an estrogen receptor, and we have an antagonist bound to the receptor. So you can see there's antagonist molecules here and here as well. Now, not all receptors look the same. Receptors can look quite different, but they all transverse the cellular membrane. And what we're going to look at later on in this section is how we can design agonists and antagonists, so molecules that either activate or prevent the activation of the receptor, and we'll have a look at their molecular design and the important features that these molecules need to act as an agonist or an antagonist. So how are receptors activated? Well, the receptor surface is a very complicated shape, and this contains selective binding sites for chemical messengers. Now, the theory behind receptor activation is very similar to the concept of an enzymatic reaction in an active site. And the key difference between a substrate binding into an active site of an enzyme and a messenger binding into the receptor site on a receptor is that the messenger molecule binding into the receptor does not undergo a chemical reaction, but a substrate binding into an enzyme does. So what does happen then? What happens when our messenger molecule enters into the receptor binding site? Well, the binding of the messenger to the receptor results in an induced fit. So the messenger molecule enters into the binding site, forms lots of intermolecular interactions with functional groups within the binding site, and draws those functional groups closer to it. This then causes the receptor to change shape, and it leads to transmission of the message through the receptor. And so this is shown on this diagram here. So here we have a messenger molecule that can bind into the binding site on a receptor. Now this receptor is of course embedded in the cell membrane. Now when the messenger enters into the binding site on the receptor changes its shape, so it has an induced fit, and this triggers the sending of the message to the inside of the cell. Now once the messenger has done this and triggered the message to be sent, the messenger molecule is then released from the receptor. Now this is a very important point. The messenger molecule needs to be released so that we do not permanently activate the receptor. Just like with enzymes and permanent inhibition, permanent activation of receptors is not something that is desirable. And so therefore we can consider this just like the permanent inhibition of an enzyme molecule. We don't want to permanently inhibit our enzyme molecules. We also do not want to permanently inhibit our receptors. However, again, note the importance of the induced fit process. It is this induced fit that is induced by the binding of the messenger molecule that allows the message to be sent from the outside of the cell to the inside. So how does a messenger molecule bind to the receptor and change the shape of the binding site to cause an induced fit? Well, if we consider a hypothetical neurotransmitter and a receptor, what we can see here is on the neurotransmitter we have several different binding regions. So for example, we have a region with a positive charge that could have an electrostatic interaction with a group in the receptor that has a negative charge. We also have a group able to act as a hydrogen bond donor or acceptor, so this OH group here. And we also have a 6-membered aromatic ring that would be able to bind through a van der Waals binding interaction. Now if we look at the receptor contains the complementary functional groups to the neurotransmitter. So we have a region that is able to act as a hydrogen bond, donor or acceptor, a van der Waals binding region, and an ionic binding region as well. Now this means that the neurotransmitter and receptor have 3 potential binding interactions that they can adopt. And if we put the molecule into the binding site, we can see what happens. So within the binding site, the 6-membered ring has a van der Waals interaction with the van der Waals binding region on the receptor binding site. We also form a hydrogen bond between the receptor and the neurotransmitter. And we also have an electrostatic interaction between the neurotransmitter and a group within the receptor as well. Now these binding interactions serve to pull the binding site in towards itself and towards the neurotransmitter, making it smaller. Now this change is called the induced fit, as you already know, and it doesn't need to be a big change to the overall structure of the receptor. It can be a very small change and still transmit that chemical message to the inside of the cell. Now this is actually an example from a lecture that you'll see in part 2. where we're looking at the mechanism of action of medicines used to treat cardiovascular disease. Now in grey we have salbutamol, which is an agonist for the beta-1 receptor, and in cyan, cyanopindolol, and this is the antagonist in the beta-1 receptor. Now this structural overlay shows the difference between the two, and so the structure in grey is the activated form of the receptor. It has been activated by the salbutamol. In cyan is the inactive form, and you can see that these structures are still incredibly similar to each other. Now if we have a look at the right hand image here, what we can see in grey is the salbutamol position overlaid with the cyanopindolol in cyan. And in fact here we can see that the position of the binding groups is conserved until we get to the rings. And we can see that the cyanopindolol is larger than the salbutamol, and this is because, as we'll cover later, antagonists are trying to prevent the induced fit at a receptor. Agonists are trying to have that induced fit, antagonists are trying to prevent it. And so the difference in positions of the atoms here are very, very similar. There's a very small difference between the two, but that difference is enough to give you the difference between activation and inactivation at that receptor. And so we're going to start off by looking at three main types of membrane-bound receptors. These are ion channel receptors, G-protein coupled receptors, and kinase-linked receptors. And then in a later section we'll look at the design and function of agonists and antagonists against these different types of receptors. So first of all we're going to look at ion channel receptors. Now ion channel receptors control the movement of ions across the cell membrane, so they allow ions to move in and out of the cell. Now these are made up of protein subunits which transverse the cell membrane. So here we can see that the protein itself moves through the cell membrane. It holds open this gap, and each of these sections is a different subunit of protein. Now all of these subunits have joined together to produce our ion channel. Now inside this ion channel we have a hydrophilic region. So you can see this here. We have a hydrophilic tunnel that allows ions with a charge to move through the center of this protein complex. Now a lock gate controls movement through the channel. And so here we have a messenger molecule which can bind to our ion channel. Now our ion channel without the messenger molecule is closed, so this lock gate prevents the entry or exit of ions from either exiting the cell or entering the cell. However, when the messenger binds into the receptor binding site, the induced fit of the ion channel opens this lock gate. And when this lock gate is open, ions can travel freely in and out of the cell via diffusion. So this messenger molecule changes the shape of the ion channel and allows it to open, allowing the ions to diffuse in or out of the cell. Now this is what a potassium channel actually looks like, as a diagram which shows you both the beta sheets and alpha helices present in this receptor. Now the alpha helices are shown in red, and you can see that most of them are down near the base of the receptor, with the beta sheets in yellow. It's this section here in red that traverses the membrane of the cell. The structures in yellow up here, or predominantly in yellow, are beta sheets which sit on the surface of the membrane. But it's this section down here with the alpha helices that traverses the cellular membrane. Now in fact you can see in here three metal ions that are in the middle of this receptor in this particular structure. And so this is the space that will open when the receptor has an induced fit and allow these metal ions to pass through. At the moment the receptor doesn't have a messenger molecule bound and the lock gate is closed, and that's why these ions are trapped at the entrance to the receptor. Now we can also get ligand-gated ion channel receptors. Now these are ion channels, but they don't contain a lock gate. Instead, they contain a much more complicated mechanism. And this mechanism is opened by the binding of small numbers of neurotransmitter. Now this binding allows for a very large change in the ion channel, and it facilitates ion transport across the membrane. Now the binding site for the neurotransmitter in ligand-gated ion channel receptors is completely different to that from a lock gate. Now it requires a very complex series of events to open the channel, and so it is considered a separate subtype of receptor. Now in this case the lock is constructed of 5 kinked alpha helices. Now these all rotate when the neurotransmitter molecules bind, and they allow the ions in and out of the cell. So what does this look like? Well, here we have our ligand-gated ion channel receptor, and we have the 5 kinked alpha helices which transverse the length, or rather the width, of the cellular membrane. Now if we look down from the top of these helices, what we would see is a closed gap. So we would see 5 transverse helices and an area that is closed. Now when the neurotransmitter binds, all 5 of these alpha helices rotate, and that then converts the ligand-gated ion channel receptor from a closed form to an open form, allowing ion flow through the receptor. Now in comparison, a lock gate is much more simple, it's much more straightforward, and so as I've said, these are considered a different subtype of an ion channel receptor. So now we'll look at G-protein coupled receptors. Now these are receptors which respond to hormones and slow-acting neurotransmitters. Now a G-protein coupled receptor consists of a membrane-bound protein, and this protein then activates membrane-bound enzymes through a process called G-signaling. Now the mechanism of activation relies on the induced fit of the messenger changing the shape of the protein. So let's have a look and see what this looks like. So this is a G-protein coupled receptor here. Now this G-protein coupled receptor on the outside of the cell will have a receptor binding site, and on the inside of the cell will have a site that G-proteins can bind into. Now this site is closed as long as there is no messenger in the binding site on the outside of the cell. When the messenger enters into the binding site on the receptor, the internal binding site on the inside of the cell opens, and a G-protein is able to bind into this. Now when this happens, the G-protein is processed, and G-protein subunits are released into the cell. Now these go into the membrane and activate membrane-bound enzymes that are on the inside of the cell. Now the binding of a G-protein subunit into the enzyme then opens the active site of the enzyme and allows the enzyme to be activated. So it allows the enzyme to start taking in substrate, processing it, and releasing product. So in this system we have a messenger that binds into the binding site. This opens a binding site internally in the cell for a G-protein. This protein is then broken down into subunits which can then travel back to an enzyme present within the membrane of the cell. This enzyme is then switched on by the binding of the G-protein and allows for the processing of substrate into product by that enzyme. So essentially in this situation we have a two-stage messenger. We have our external messenger, which is our molecule that binds into the binding site on the outside of the cell, and then we have to process G-protein to act as a messenger inside the cell to activate membrane-bound enzymes. And both the activation of the receptor and the activation of the enzyme relies on induced fit. So for the receptor, it's the induced fit that opens this binding site and makes it available for G-protein to bind into. In the enzyme, it's the binding of the protein subunit at an allosteric site that induces an induced fit on the enzyme and allows its active site to open up and process substrate into product. Now the last example we're going to look at is that of a kinase-linked receptor. Now this is a receptor which can activate an enzyme directly. Now this is because the receptor contains both a receptor binding site and the enzyme active site in it. So what does this look like? Well, here we have our kinase-linked receptor here. We have a binding site on the outside of the cell for a messenger molecule to enter into, and an active site on the inside of the cell. Now when our messenger molecule enters into the receptor, we get an induced fit, and this opens the active site that is present on the outside of the cell. This active site can then begin to take in substrate and produce products through enzymatic catalysis. So this receptor has both a receptor and an enzyme built into it. The receptor binding site is on the outside of the cell, and the enzyme active site is on the inside. Now hopefully you've seen that receptors are very important in transmitting messages into cells, and it's for this reason that malfunction of the receptors can lead to a variety of health problems. Now malfunction could be because of either increased or decreased levels of neurotransmitter or hormone, and so we can produce two different types of drugs to help control this. We can produce agonists. Now these mimic the natural neurotransmitters and activate receptors. Or we can produce antagonists. These block neurotransmitter binding and deactivate the receptors. And we'll look at both of these in the next sections.